Thanks for tuning in today. I'm Dr. Nick, the Incrementalist. Join me as I seek out the small incremental changes being applied in other industries that we can learn from and that can be applied in healthcare. Can these changes bring immediate value, but also add up to the big improvements and revolution we need in healthcare? Come along with me to explore the possibilities. My innovative guests from around the globe have used small incremental improvements to achieve their moonshot.
And today I'm delighted to be joined by Michael Hufford. He is the CEO and co-founder of Lygenesis. Michael, thanks for joining me today.
Oh, thanks for having me. It's a real genuine pleasure to be here.
So for the benefit of the listeners, I think it's always important to get a little bit of background. Tell us about yourself and your journey to this point and how you got here. What happened?
Well, frankly, I'm incredibly fortunate to have been born in these interesting times, right? As the old Chinese proverb goes. So I, very brief background, right? I did my undergraduate work at Purdue University, got a master's and PhD at the University of Pittsburgh before completing a clinical and research fellowship in the Department of Psychiatry at Harvard Medical School. Was a professor for a short time, but got recruited to my first startup. Over 20 years ago now, before such things were fashionable, and got exposed to really the consulting side of the pharmaceutical industry. So at the time I was doing a lot of patient-reported outcomes consulting, but got involved in a lot of FDA meetings and scientific advisory boards. And then from there had just been very fortunate to work in a number of different really fascinating areas of drug development. So I worked on xenobiologics at Amylin Pharmaceuticals, worked on a number of products in development that eventually won FDA approval there. And it really ended up being very therapeutically agnostic in terms of the areas that I worked in. But what I ended up specializing in was really that value inflection you get with positive proof of concept data in humans, right? So really helping assets get through the FDA process and giving them the key test to either go or no go, right, in that phase 2 trial. And so that ended up being a particular area of expertise of mine. And I was just very fortunate a few years ago to meet my co-founders that would go on to form Lygenesis. And it so happened that that was the hurdle facing them. So I was the one who had sort of crossed those hurdles in the past and just thrilled to be able to help them move forward now.
So it sounds like from your past that you actually like all of this regulatory oversight, navigating through that process. I'm serious. I mean, it's you seem to relish that whole experience of driving the science through a process.
It's funny you say that, right? Because my experience of almost all FDA interactions is one of, it's typically a gradient of frustration. But that being said, I think you're right that what drew me to business outside of academia, frankly, right, was this intersection of science and business. And at the end of the day, when you're developing, drugs for human disease, you're of course dealing with the FDA. And I was fortunate to work with a number of chief medical officers and chief quality officers that I guess I came to understand that just the way you tell a scientific story to investors, right, to get them to invest in your technology, it's a different type of storytelling, but you're still really telling a scientific and quality story to the FDA to reassure them that you're not putting patients at risk, using good listening skills to understand what their concerns are, and then, reflecting that back in your program. And I think I've just come to appreciate the remarkable value inflection you have in developing assets that come with meeting those FDA milestones, right? So as an entrepreneur, right, when you have those FDA meeting minutes that you can put up in front of a venture capitalist and say, look, I know exactly what my last trial is going to be before my first in human study, because here I have the minutes from the FDA meeting. That becomes so definitive and in many ways kind of stops you kind of chasing your tail, right? Like very often in fundraising, you're sort of always chasing the next milestone and you kind of can feel like you're on a hamster wheel, never making progress. But when you have those FDA meetings, it really can help say, look, you're in or you're out. This is what we're doing. We're moving forward. Get on board because this is what we're doing. I've had the good fortune of being able to leverage that both in my fundraising for my companies in the past, and then also with corporate exits, the same thing, right? It's having those definitive FDA milestones either lets you decide to stop investing in an asset when things don't work, or conversely, really gives you fuel for the fire when it does. So I guess I would say I'm not sure I enjoy the regulatory interactions per se, but they are sort of at the fulcrum of either moving investors off of what's that great phrase I heard that investors live on a teeter-totter between fear and greed, right? And your goal as an entrepreneur is to kind of shift that teeter-totter from fear to greed, right? And it's a bit unfair, but not entirely, right? So I think those FDA meetings can serve as a fulcrum to kind of pry investors to get them engaged. And conversely, when you're dealing with large pharma partnerships, right? Having those FDA minutes, it just eliminates all of the guesswork. So instead of a clinical team saying, oh, but the FDA might require you to say this or that, you can say, hey, I've met with them. We had a meeting. We discussed just this. This is what they said. And it just becomes a catalyst, I guess, right? And that's how I see the regulatory interactions as a necessary catalyst, either to advance your fundraising objectives or to advance your kind of business objectives on the partnering side.
So before we get into Lygenesis and the origin story, I have to ask you, what's your superpower with regards to investing in the FDA? Because it sounds like you have it. There has to. It's not a secret, but there has to be something about your approach to this. Because what I get very clearly from the way that you describe it is not necessarily love, but absolutely admiration, respect for the process and the way. But I know many people struggle with it, but you seem to have navigated this and adapted. Do you have any small thing that you could give in terms of insights as to how to successfully navigate that space?
It's a great question. It certainly doesn't feel like a superpower. I appreciate the compliment. I would say, I joke sometimes, but I think it's true. So, I got my PhD in clinical psychology, and so I have always had a kind of soft spot for the CNS indications and psychiatric disorders, certainly. But among that training, right, you get a tremendous amount of training of listening and being empathetic, right? And so while I did a very research-oriented program, is where I kind of focused on research. So that clinical training is valuable, right? Because I think a lot of times it's easy when you're meeting with the FDA to get very frustrated for reasons I'm very sympathetic with. But if you think of things from their point of view, right? They're looking at all these sponsors. They see what happens left unchecked, right? Patients can be put at risk. So I think regulators obviously play a key role. And if you listen to their concerns through and try to take their point of view and craft your regulatory documents, like folks joke about speaking regulatory, but like a lot of it, right, is writing in such a way that you're connecting the dots and you're empathetic to your audience, right? So that, and I've been in meetings that folks don't listen well sometimes in FDA meetings, right? And I think, meetings can be won or lost in your reaction in real time to the feedback that you get. And so, not being defensive, at least trying not to be defensive. I don't know that I always succeed, but, trying not to be defensive in those interactions, to understand where the concerns are coming from. And conversely, when you work with your scientific colleagues. So, I have a great fortune to work with some brilliant, both, academic physicians as well as academic researchers. And very often, I think folks get tripped up because you're not speaking the same language. And so it's a matter of almost translating. You're trying to translate, on the one hand, the deep science into something that's digestible to a regulator that isn't deep in that science. Conversely, you're trying to translate the regulatory concerns around patient safety to your colleagues that may not understand kind of where that's coming from, right? Understand it intellectually, but not always sort of the form it takes from the FDA. So I guess like a lot of what I see myself as doing is translating and being a proactive translator. And I think if you do that well, you can engender a lot of trust, and that reflects itself in the requirements in your program and everything else. So I think it's just oftentimes a breakdown of folks talking past each other in some of those regulatory interactions. And look, the FDA doesn't make it easy, but I also, tell my academic colleagues, imagine trying to run an organization where you're always routinely thousands of FTEs down from where you need to be. You're always under budget. You're always overworked. You can't pay people what you'd want to pay them, right? I mean, those are real challenges to running that agency. Absolutely. And so you just have to approach it with an appreciation that, man, these people are doing the best job they possibly can under a crushing workload, chronically short-staffed. And in that ways, right, I think in many ways resemble their academic colleagues in some respects, right, where you're oftentimes understaffed and always fighting for the next grant and what have you.
Yeah, I think fantastic insights and, helpful to others to sort of think about it in those terms. And, the other element to this is that, from that lens and perspective for the FDA, ultimately, approving something to be used, they don't get quite the same cue points as, preventing a disaster from occurring. And, that's hanging over them all the time. And I think people forget that. So fantastic. But we need to get to Lygenesis.
Sure, yes.
Lygenesis is this incredible technology. So help people understand what it is. Summarize what Lygenesis is and how it came about.
Sure. Well, look, at the highest level, Lygenesis is a cell therapy company that uses the lymph node as an in vivo bioreactor to regrow a functioning ectopic organ. And really what that literally means is that we use the natural biology of the lymph node as a bioreactor, naturally meant to bioreact T cells, right, to help you fight infection. We wouldn't be here if it wasn't for having 400 to 500 lymph nodes as these sentinel bioreactors spread throughout our body, right, to help fight infection. So these things have evolved to be incredibly efficient at developing T cells to fight infection. Of course, cancer hijacks that biology, right? Cancer, the lay public's aware that when you get cancer into a lymph node, it's a bad thing. And it's a bad thing precisely because that is both a conduit for its spread, but also a conduit for the formation of tumors, because those bioreactors are so efficient at growing cells. And the fundamental insight from our chief scientific officer is that biology could be leveraged to a therapeutic effect. So the way cancer hijacks the biology to form a tumor, Dr. Eric Lygenesis, our chief science officer, realized that, over a decade ago now, that same biology could be used to regrow functioning tissues. And he started with the liver. It has since now expanded. So our platform includes the ability to regenerate a ectopic liver, but also a pancreas, thymus, and kidney. But taking the liver as one example, what Eric Lygenesis showed in his laboratory, starting with small animals, going through to large animals now, and starting our first clinical trial here soon, is that if you inject hepatocytes, liver cells, into lymph nodes in the abdomen, over time, those hepatocytes are very happy to be in the lymph node. They find it a very hospitable environment. And in the context of a diseased liver, when the liver is emitting dozens upon dozens of biochemical signals trying to get hepatocytes to regrow, but an advanced liver, right, with fibrosis, say, has become a very inhospitable environment. So these hepatocytes in lymph nodes detect those signals, begin to engraft, proliferate, vascularize, and eventually the lymph node disappears. And what you're left with, we have some great pictures of this in our Nature Biotechnology publication, are these ectopic livers that exert life-saving effect in animals that would otherwise die in a matter of days or a few weeks from, in small animals, a genetically induced disease called tyrosinemia type 1. That we can rescue those animals from otherwise fatal liver disease. So that was Eric's fundamental insight a decade ago. And to his credit, he doggedly pursued that line of research he studied almost 400 mice by the time I had met him. He'd done 2 replicate studies in pigs, one at the Mayo Clinic, one at the University of Pittsburgh, in different models. And literally, when I met Eric, you get a chance, I was an entrepreneur in residence at the time, you get a chance to meet a lot of brilliant academics. And as an academic researcher, to his credit, he kept trying to get it not to work. And he literally couldn't get it not to work. The cells engrafted over and over again because the environment of the lymph node was so hospitable, right? And so it was remarkable that by the time I met him 3 years ago, he had done the majority of the IND enabling work really without meaning to, without intending for that regulatory purpose. But by the time I met him, I'll never forget our first meeting. The hairs on the back of my neck stood up as, he showed me the mouse data. It was incredible. These great pictures, just jaw-dropping stuff. And I thought, look, that's fantastic. What about large animal work? Fully expecting him to say, oh, well, we've not gotten to the large animals, or, it didn't work. Instead, he's like, oh, yeah, we've got 2 replicate studies in pigs. We rescued those. We rescued tyrosine pigs. We used a portacable shunt, which is a surgical procedure to mechanically induce liver injury. We rescued those pigs, And so you just think, holy cow. This is, as you well know, right? So often in drug development, you're pushing against biology. You're trying to either trick the body or get it to do things that it doesn't naturally want to do, right? So you're always dealing with unwanted side effects or, off-target effects. And here it was really the opposite. It was like the body wants to do this. We can't get it not to. If you get the cells in the lymph nodes, the biochemical signaling is so powerful, right? The Greeks realized 5,000 years ago that the liver would try to regenerate. That was, you may recall, the myth of Prometheus, right? Prometheus, the legend, right, stole fire from Zeus, gave it to man. And so Zeus punished Prometheus by chaining him to a rock. And the Greek myth was the eagle would come and eat his liver every day, and it would regenerate. And then he'd be tortured by having the eagle come and eat his liver again. So I joke with folks, the Greeks had it right 5,000 years ago. The liver tries to regenerate. It was this fundamental insight that we were able to use the lymph node as an in vivo bioreactor that has kind of made that a reality today.
So for those of you just joining, I'm Dr. Nick, the Incrementalist, and today I'm delighted to be joined by Michael Hufford, the CEO and co-founder of Lygenesis. We were just covering the incredible concept that you essentially described, the potential to turn our own bodies, in this case, the lymph nodes, into actual organs that, we have tremendous challenges with in healthcare. We understand the concept of transplants, but we have this massive shortage.
Yes.
For the benefits of those listening, just clarify where that stands in terms of, lost opportunity of available organs.
Well, there's really dual tragedies when it comes to the transplant list. The first tragedy that people are aware of, that there's more than 100,000 people in the U.S. currently on the wait list for a transplant, right? So there's the supply-demand imbalance is absolutely profound. There is far more demand than there is supply. And that's true whether you're talking about hearts or livers or lungs, kidneys. Some wait times are longer than others. I think the average wait times for a kidney is almost 2 years now. Liver is still under a year, but you're on the list a very long time. And so, look, the reality of that means dozens of Americans die every single day waiting for an organ that never comes. But I said it was dual tragedy. That's the first tragedy. The other tragedy is what a lot of people don't realize is to even qualify for the transplant list means that you have a lot going for you, right? And so in some diseases, advanced liver disease being a good case, it can be a 1-to-1 to 2-to-1 ratio of the patients that don't even qualify to make the list. So the double tragedy is that while many Americans are aware and hopefully sign up to be an organ donor, which they absolutely should, I think, the fact is many patients are so sick because of either cardiovascular comorbidities or past cancer diagnoses or whatever the specific indication is for that organ. The fact is many patients never even make the list, right? And so at the highest level, what we're trying to do is, hopefully one day eliminate wait lists altogether, at least for some of these organs. And I think, our technology, because, today a single donated organ can treat one patient. Using our technology, a single donated liver, taking that as an example, has enough liver cells, these hepatocytes, using our technology to treat 75 or more patients. So you want to talk about, that's a complete disruption of the supply-demand calculus when it comes to organ transplantation. We engraft those cells using an endoscopic ultrasound procedure. So instead of what's usually one of the most, risky, surgeries you can have in terms of, transplant surgeries have gotten, obviously very good and don't get me wrong, very safe and reliable as time's gone on, but still a major surgery, right? And that's why many patients don't qualify for it. Using this endoscopic ultrasound procedure, this would be an outpatient procedure under light sedation, right? So you've decreased the medical risk. Today in the U.S., transplanted liver costs on the order of $800,000 in terms of the costs. An endoscopic ultrasound is billed at, $10,000, $15,000, right? And so when you think about just from a business perspective, right, the return on investment, transplant is so expensive and relegated to actually, in many cases, a minority of the patients that need it. Our technology's ability to disrupt the supply-demand calculus and engraft these cells using an outpatient, less risky endoscopic ultrasound procedure, that 1-2 punch, right, is one of the things that I'm personally most excited about.
That's just truly incredible. I mean, you just think about the amplification of the available organs, solving problems, and in fact, addressing that second part of the tragedy, the people that don't qualify would qualify for this because you're talking about I can't describe it as anything else but a minor procedure.
Yeah, I mean, and certainly relative to full-blown transplant. And my chief medical officer, Dr. Paulo Fontes, has spent his life as a transplant surgeon and worked at the University of Pittsburgh and the Starzl Center, has treated thousands of patients. He personally was the one who would tell patients, look, I'm sorry, your loved one's never even going to make the list. If they made the list, he was the one who might transplant them, right? But giving those patients some hope, right, is something that very much drives us forward. And as I had mentioned, so liver is our lead clinical indication. We have proof of concept data, though, in animals showing that we can regenerate kidney, we can regenerate the pancreas to potentially address issues in type 1 diabetes, as well as the thymus that has some very exciting indications, potentially for aging. As we age, our thymus involutes and, in fact, disappears over time. And so the ability to potentially regenerate the thymus has some very exciting longevity and anti-aging implications as well.
Wow. Well, let's talk about a superpower. So we've got a couple of minutes left.
Sure.
Tell us where you are and when we're going to start to see. You've had approvals. You're marching through. Give us some estimate of the sort of timeline and the next things coming up.
No, it's a great question. So we've been working very hard. We received our clearance from the FDA to begin our human clinical trials late last year. We're starting a phase 2A trial in patients with end-stage liver disease. We hope to have that first patient in here in just the next few weeks to a handful of months. So we're getting very close to starting enrollment. That's extraordinarily gratifying as a drug developer. So we're very excited about that. That 2A trial is going to be a dose-ranging trial. And then depending on the outcome of that, we will proceed with a more traditional, placebo-controlled trial with the FDA's input. But the FDA has been very helpful. They've given us some great input on the design of our 2A trial, and we're looking forward to getting started.
Wow, that's really exciting. So, starting human studies.
Yes.
You think about it from, the original insight and innovation, we're talking many, many years to get to this point.
Yeah.
But I can imagine from your perspective, this is everything that, the organization and you've been working for. I know you've been working on this for a long time.
Yeah, 3 years in, we're incredibly gratified. Can't wait to get this to the bedside to help patients. And you couldn't ask for anything more than the exciting science, great colleagues, and we've had great engagement from the FDA and very supportive investors. We've had Juvenescence, Longevity Vision Fund, a number of folks have been early supporters, continue to support us. So those folks have been instrumental as well.
Fantastic. Well, as usual, we've run out of time, but I think I can't help but be excited about this. I'm fortunate I don't sort of live in the world, but I've certainly experienced and participated in that world. I've seen the disappointment. It is an astoundingly difficult problem to address because we know what to do. We just lack the raw material. And here you are creating amplification for the raw material. So just excited to watch this and very grateful that you were able to spend the time and share the details. So thanks for joining me on the show, Michael.
Oh, thanks so much. It was a real pleasure.
Thanks for joining me today. Do you have any better ideas, or have you found a small incremental change that's brought about a big improvement in your world? Let's continue the conversation on our hashtag, The Incrementalist, or share with me at Dr. Nick1 on Twitter. You can find more information about the show on our program page at healthcarenowradio.com. And tune in next time to hear my discussions with leaders and innovators from around the globe who've revolutionized their space by using small incremental improvements to achieve their moonshot. I'm Dr. Nick, the Incrementalist, and I'm starting a revolution through evolution.